Well, good morning, everybody. Welcome to another Chatting with NDGS Paleo and Friends, our COVID-19 edition. Today we have a guest speaker who's talking with us all the way from across the seas in Berlin. Her name is Yara Haridy, and she got her bachelor's and master's at the University of Toronto. And she's currently working on her PhD over at the, I'm going to say this wrong, Museum für Naturkunde. I probably said this terribly. in Berlin, Germany. So she's going to be chatting with us about pathologies and bones. Now, before we get right into that, most of my specimens are sitting at work, but I do have a couple of casts that I made from my undergraduate days with me here, one of which is this beauty. This is part of an Edmontosaurus, or a duck-billed dinosaur rib bone. And you can see it's nice, pretty smooth bone here, nice, pretty smooth bone here, and then something happened in the middle. It's just this big, huge bump or a callus sitting in the middle here. So something happened, I don't know what. And then we have, I have a little backbone here. This is a caudal vertebra or a tailbone, also from our Edmontosaurus. And it has some extra holes in there. Let's see if we can get the camera to focus. Focus. There we go. It has some extra holes in there that should not be there. And it's all gnarly. It's all bumpy. It's had some kind of terrible malady running through here. So maybe we can find out a little bit more about those too. But for now, welcome, Yara. Thank you so much for joining us.
Thank you so much for your intro. I'm really excited to get to talk about this. So I guess I'll just jump in. All right, everybody can see this, right? Yeah, I'm really excited to get to talk to you guys about paleopathology and bone evolution. Overall, I'm hoping to convince everybody that things that I actually study, the things I actually study for my PhD are actually related to paleopathology. So I mostly study bone evolution and basically bone cell evolution. And paleopathology has basically been a side project or a side endeavor that was just always really, really fascinating to me. So because of that, I've tried to relate the two. So let's get into it. So first, a little bit about me. I'm a big fan of communication. So go ahead and follow me on Twitter if anybody isn't already. This is where I usually yell bones and fossils and what they do for science. And here's a few ways I like to do that. I used to like go into collections when we were allowed to and bring out fun specimens and show people what they actually mean and what they tell us. On the left here, I have a velociraptor puppet, and it's basically one of the projects that we're working on soon in a way to show like flight evolution. Other things I work on, I basically don't discriminate when it comes to fossils. I study both the big and small. As long as it's bone, I'll study it all. It's kind of what I like to say. So anyone that has any weird pathologies, they can throw it my way. So let's actually get into it. So bone cells and pathology, and how do they actually relate to each other? And that's what I hope at the end here you guys will actually agree with me on this, that they're actually intrinsically involved in one another through bone evolution. So what I actually study is bone cells in the fossil record, except of course cells don't make it. They don't fossilize well, but what they do is they leave impression. Basically leave these like beautiful caves called osteocyte lacunae. So osteocytes are bone cells, and lacunae just literally means space or cave. And they leave this cave that basically is the exact same shape as the cell. And they're in the bone and really pretty fossils, and when preserved well and not crushed, this preserves quite well, so we can actually figure out what the cells looked like. So one of the ways we study that is histology. We cut up bones. I'll get back to that later. And from there, we can figure out what bone cells looked like, how they're connected, what animals have more or less. And that's how we study osteocytes, so these bone cells. Now, of course, bone cells are more than just what's preserved. We have the osteocyte, which is the one that we can see in fossils. We have osteoblasts, the ones that actually build bone, and they don't preserve in fossils because they survive on the surface of fossils, so they don't fossilize or survive on the surface of bones. They're what actually excrete bone. So they basically push bone downwards and they can become osteocytes, but they don't preserve. Osteoclasts, the ones that destroy bone or remodel, and osteoprogenitor, all the cells that basically become an osteoblast. So they're the pre-osteoblasts, basically, and these also don't preserve in fossils. So the only ones that really fossils are osteocytes. This is a very, very small window. And osteocytes haven't always been around in bone. They actually evolve later on, or show up later on in jawless fish, and then basically throughout until some fish secondarily lose them. So then that brings up the question of, well, if they show up sometimes and not others, and if some of the earliest vertebrates apparently don't even have them, then what was the point? What was their function? And these are some of the questions that are really hard to answer in evolutionary biology and really can't be answered except with fossils. However, with fossils, we have this sort of black box. We don't really know what these cells were doing because they're not around anymore. We have, if the bone is perfect and it's formed perfect, then we just assume, okay, the cells are there. They're doing everything that modern cells are doing. And we can't really say much beyond that because the bone was made right and that's all we got. But this is where paleopathology comes in. And this is where I love this quote. We study rare events to understand common occurrences. The storm of the century to understand how weather works. A world war to understand how normal behavior, human behavior works. A mutant to understand the process of normal growth and development. Now, of course, in developmental biology, biologists use null models. So they basically have mice or whatever the experimental animal is, and they can knock out a certain gene and they can figure out how something works because they knocked it out. But when it comes to animals that are extinct and we don't have these null models, we don't have these knocked out versions, we can't study that. So that's again where something called paleopathology comes in. Or pathology. It's the study of ancient diseases, a deep time perspective on the origin and evolution of disease. And basically pathology just means anything abnormal. It can include injuries, it can include deformities, it can include actual like ailments, diseases from viruses, bacteria, etc. We'll get to that in a sec. So here we have an example of an osteosarcoma of an early hominid, and it's the earliest example of cancer in humans to date, I think. And on the other side, we have the earliest pathology in amniotes, actually not vertebrates, but early cancer in, so this is another osteosarcoma, so that's bone cancer. Now, the question is basically, that's great, you have bumps and lumps on old bones, but what does it actually mean? What can paleopathology tell us? What can we really diagnose a fossil? Can we actually diagnose a fossil? And what does it matter? Is it just geoscience? And what about the big picture? So these are kind of the questions that I'm always thinking when someone brings me a bumpy, lumpy fossil. We can diagnose the animal, sure, but can we tell more from this? So let's go through the kinds of pathologies, and this might help you all at home if you have some weird, bumpy, lumpy fossil to actually put it in a category. So the most common that you'll ever find are traumatic pathologies, and that basically means an animal broke its bone or got bit or something as a result of a trauma. And that can be a break, that can be a bite, that can be anything at the initial like interaction, whether it's animal on animal or animal with its environment. So that's a traumatic pathology, usually a break or a bite. And we can tell that these happen because they can heal over quite well. There's developmental pathologies, so basically the animal grew in an incorrect way or a non-typical way. Sometimes they don't actually affect the animal at all, sometimes they do. There's additive pathologies, so things that are growing on top of other things that shouldn't be, basically. Erosive pathologies, so things that are taking away from the original structure of the bone. And neoplasms. So that's a change in the type of tissue. So basically, if you have bone and it's growing into cartilage, or there's an addition of cartilage, or all of a sudden it's growing muscle where it shouldn't be, these are neoplasms. And neoplasm and cancer is a type of neoplasm, but not all neoplasms are cancer. Now, this is a question that everyone loves to ask me, which I think is kind of fun. How do you know if your fossil is sick? Basically, I ask them this. I tell them to do the simple: look at the bones, because that's all we have when it comes to animals. So, of course, when it comes to extinct animals, we have a very biased look at how the animals could have been sick. So, anything that we analyze or anything that we find in fossils is already only things that left marks on the bone. So let's take a look. Here's a good example from our own collections at the Museum for Naturkunde. And this is a sauropodomorph, and this is their tail vertebra. So this is one I got to find when I was just rummaging through collections. And this is one of the obvious ones, right? You look at the series of vertebrae, they look upset. They don't look comfortable. The bone edges are really irregular. They kind of like mush together, but don't want to be together. And then there's obviously the fused vertebrae in the middle. The hemal arches, the little spines that come down, are fused to the actual vertebral body, which is abnormal in this case. And we took a look at the rest of the skeleton because we actually have the complete skeleton of this individual, and the rest of it looks pretty fine. So it was a very localized pathology, but very obvious. So one of the very first things I always tell people to do if you're trying to diagnose your fossil is you look at it. So this is obviously pathologically fused, and there's a bunch of other odds and ends that are that are problems with this fossil. I thought it was a cute one. Yeah, there you go, just in case you missed it. So, and again, so just look at the bones. Usually that's the first way that we diagnose fossils, or that we even notice that there's a problem. So here's an obvious one. I think this one's from the LACM. And I think it's a cast. But yeah, you can tell. Again, these ribs don't look right? They don't float. The alignment is incorrect. And then there's obviously this massive growth. So this is likely a traumatic pathology, like what we talked about the first way, which is ribs actually get hit a lot, especially with large animals. A fall can cause a problem if they're fighting or hunting or whatever. Ribs are just very exposed. They're thin spicules of bone that are on the sides, so they tend to actually accumulate quite a bit of traumatic pathology. So they're a really good place to look if that's what you're looking for. But most of the time it's just traumatic pathology. So it's something hit, and then you're trying to figure out how the animal healed, or sometimes it doesn't heal well, sometimes it heals perfect, never sick. So the weirder ones, in my opinion, are developmental deformities. So these ones are from the giant amphibian Metoposaur from the late Triassic. And while it just kind of looks like a lump of bone at the beginning, you actually take a closer look and you realize, oh, there are several fused vertebrae, but they're not just abnormally segmented. So during development, basically your vertebrae will segment into the proper cell clusters before they actually start to form the bone. But if that segmentation doesn't happen properly, then you can get these weird fusions and things that are called block vertebrae, which is basically several vertebrae that didn't ever actually separate. You can get wedge vertebrae, which is incomplete separation. So all kinds of things like that that point to how the animal developed, but basically misdeveloped or abnormally developed. So you get these interesting deformities. And they're quite common in amphibians, actually, but not so much in other tetrapods or amniotes. Here's another one with where you can see that there's an articulation of where the ribs are. But again, so they're not actually spaced. There's probably what should have been 3 vertebrae here are now squished into this one block vertebrae. It's kind of amazing that this kind of stuff preserves. So one thing that looks abnormal, right? And you have probably put it in one of the categories, whether it's a traumatic or developmental or neoplasm or additive or whatever. You can only tell so much externally. because a bumpy, lumpy bone could be so many different things. So this is where you start thinking, how do you actually diagnose it? How do you get more out of what this pathology is? And my go-to is usually histology, if the curators will let me, because most people are very precious about their fossils, which is fair. But honestly, it's such an amazing method. And of course, as a histologist, I'm super biased, but here's a good example. So this is basically a lump on a parareptile that we don't know which one exactly, but we think it's an ulna of a parareptile, and there's just a lump on it, right? So at this point, that lump could be anything. It could be a partial fracture that healed, or it could be cancer, or it could be an infection. It's really, really hard to tell externally. So then when you actually section it, you can see, okay, at least the internal bone is not eroded through. So the original bone hasn't been basically destroyed by anything. So that doesn't really point to an infection. Usually anything with an abscess or any sort of infection will destroy the old bone before creating more bone on top of it. So this looks like some weird additive pathology, and we're actually still not sure exactly what had project to decide for a minute, but this just speaks to the level of detail you can get from histology. However, of course, like I said before, not everyone's ready for me to cut up their precious fossils, especially if it's like a one-of-a-kind thing that has a pathology on it, right? So once you cut it up, it's cut up forever. So that's not always possible. So the next step, let's see if we'll get to play. This is an Ice Age frog, so not that old, but it had a big lump, basically, again, bumpy, lumpy bones, and you can't really tell what it is at first. I'm gonna pause it here because, so frogs like to fuse their bones, and that's basically because of their extreme mobility. They have an intense jumping lifestyle. So when you have your bones fused together, that's actually quite normal for frogs. But this lump is not normal. So when you look externally, because we don't have the other end of the bone, it's hard to tell, okay, is this a break or is this a growth? Is this an overactive like callus? There actually isn't too much work done on early or in general on amphibian healing. So it was really hard to tell initially. And then when you scan it through, here, sorry, you can actually start to see that, yeah, it is a callus and it is a bone break that has healed because you can see the two sides of the bone and then they're covered in spongy tissue that's kind of just used to hold the two bones together. So that's actually very similar to the way you and I heal our bones. Our bones will basically make this shell around the two broken ends and then try to dissolve everything in the middle and rebuild it again. So basically this died and fossilized at a stage where it was just stabilizing the break. Again, so if a really good way is CT scanning. So this was back when we CT scanned Pathologies. Pathologies is a stem turtle. So it was really fun to scan this fossil because we all had our guesses and everyone had bets going on what the bump and lump actually was. And we were very excited to be confused, basically, because the less we know, the more we want to know. And at that point, we were just looking for, okay, what's fit this? And the way we do this is basically we turn to medical literature because we're so limited in the vet literature where not that many people will actually write about like the terminal illnesses of certain, animals, whether they're pets or whether they're wildlife. So we end up actually having to turn to human medical literature. However, we also understand that like human physiology and human metabolism is not the same thing as probably what a stem turtle had. So you kind of have to see, okay, well, if there's a lot of remodeling here, maybe that this animal would have less, and you kind of try to adjust whatever human symptoms would occur to a different physiology. So you always have to keep that in mind. But basically the way we diagnose this weird bumpy lumpy bone is we scanned it. Here, I don't know if you guys can see this, but basically if you look really close, the old bone in a slightly lighter gray it hasn't really been disturbed. So there aren't these like massive pits inside it. There hasn't been a whole, so it's a lot of, and the addition has the spiculate shape, which is characteristic of bone cancer. So it's this rapid growing bone on the surface without disturbing the initial underlying bone. And that's basically how we diagnosed it, by going through and systematically putting, okay, does this symptom exist or it doesn't exist? Is it a break, or is it a bite, or is it any of that? And we couldn't find any evidence of infection or a broken bone, like what I showed you earlier. There wasn't two ends that were misaligned. There was no remodeling. There was no evidence of bite marks, nothing. None of that was existent. So it was actually a very calm looking bone, except for this weird lump. So all these symptoms that it did have aligned with exactly what we call a osteosarcoma in modern humans. So basically just a growth of bone on top of undisturbed bone. And that's how we diagnose this paleopathology. So then, of course, when I talk about cancer in fossil record, in the fossil record, what the first thing people ask me is, so how cancer, like, you know, we thought it was something that you got from smoking nowadays, or you know, pollution or whatever. People think it's a very modern occurrence, which although the like prevalences, maybe the rates have definitely increased because of certain, you know, carcinogens, but inherently cancer is incredibly old. So we right now have the oldest record in hominids, we have the oldest record in amphibians. It goes back much further, especially when you loosen the term for cancer as just uncontrolled cell growth, which is basically what cancer is. You can actually find cancer-like things all the way down to echinoderms and bacteria and plants. There's all kinds of uncontrolled cell growth. And that's kind of really interesting because it means paleopathology can actually lead us to understanding why certain diseases exist. And this paper is super interesting. Maybe I can add a link in later. But basically it talks about how cancer, if we were going to use phylogenetic bracketing, we basically look at animals that have it today and how they're related and how far back we can trace cancer. If we look at that, we can actually trace cancer all the way down to the first multicellular organisms. And then when you think about it, that actually makes sense because for cells to actually start to proliferate together and to become a multicellular organism, there has to be rules in place. So some of the rules that these authors put in were uncontrolled proliferation, resource monopolization, regulation of differentiation, environmental degradation, and inappropriate cell survival. So if you think of these as the key rules for any cell to kind of, okay, let's make a deal, everybody, we're going to become a multicellular organism, and the rules we're all going to live by. And any breakdown of any of these rules results in cancer. So that means basically cancer is a part of our multicellular biology, and it's a breakdown of any of these rules. And it could happen at any time in the fossil record. We just haven't found it yet. So it doesn't mean that just because we found it 260 million years ago, that's when cancer showed up. But actually, that's just when we first found it. But I'm sure we'll actually find much older instances. So back to my point of why we need paleopathology, or basically things that have gone wrong to understand cells. It's this figuring out of diagnoses and figuring out how certain things heal that actually tells us, okay, if this animal here has this type of healing, then this cell has to exist. And here's a good how I actually started to look at them. So this is a normal looking vertebrae of an early reptile, a Varanopid from the Permian. And this is a less happy looking pair of vertebrae, as you can see, and all kind of bumpy and lumpy. Now, fusion of vertebrae can happen for all kinds of reasons. It can happen because of aging. It can happen for a bunch of reasons. But here, once we actually scanned it and we got to look internally, we realize there's actually a lot more going on than just fused vertebrae that can happen through age. So this might not look like it means much to most people, which is fair. But what's really interesting is there's a lot of pockets and there's a lot of pits. So if you can look in, there's basically all these giant pits of resorption. And then at the very bottom there, you can see a really thick wall of bone. So you're getting overbuilding and over-resorption. And this is really strange, and the normal vertebrae definitely did not look like this. So not only do you, but you also have a really strange kind of miscommunication between the cells that build bone and cells that destroy bone. And here we diagnose this, this mismatch is actually really well known in a lot of metabolic diseases. So basically metabolic bone diseases, one of the ways that they can occur is this miscommunication between cells that build bone and cells that destroy bone. And then that means, because we have this miscommunication, that means that that communication existed, if that kind of makes sense. So because we have the time where that communication broke down in paleopathology, now we have indirect proof that that's actually when that communication actually existed in the fossil record. Now, of course, because this is already a Permian reptile and they're always growing and we know how their bone physiology works, we already knew that that communication happened. But if you can apply this to actually much earlier vertebrates like early jawless fish, we can start to figure out how bone started to organize early on. And that's where I'm hoping to kind of take this. So if we found something similar, for example, in an early bony fish, we can tell, okay, so this communication maybe didn't exist in this form until the early Devonian or something like that. So that's some of the ways that I'm trying to use paleopathology to kind of bring our understanding of how these early cells worked. So basically, again, if we found something like that in the earliest cellular bone, we can figure out, okay, maybe all these cellular features evolved together, or maybe they evolved in a stepwise function. We actually don't know much about that yet, but I think actually analyzing pathologies in early fish will help figure out some of that stuff. So I hope you guys agree with me that bone cells and pathology actually come together and tell a bigger and more important story about bone evolution, and that pathology actually is intrinsically useful for us to tell a bone evolution story. So thanks for listening. I'm excited to hear some questions.
All right, well, very cool. Thank you for chatting with us about that. Very neat.
You're very welcome.
Yeah, so for my little Edmontosaurus rib break, probably traumatic. These are really, really common in Edmontosaurus, and sadly common in me too. I've got a few busted ones myself. Not right now, but yeah, this tailbone though, that's definitely the interesting one. I would love to have the original of this one sliced or CT scan, just because we are seeing some of that bone degradation happening. There's pits running into where the bone is.
Very erosive. Yeah.
That's really cool.
I don't know what it is about duck-billed dinosaurs. They are like covered in pathologies. They're one of the ones that, like, if you want to go find a certain type of pathology in an animal, like, you go to duck-billed dinosaurs. They're always riddled with them. I think one of the only ameloblastomas, which is like literally a tumor of teeth, is in a duck-billed dinosaur. And it makes sense because they're making a ton of teeth all the time. And, you would think that is a high, like, cellularly active area, but it's so cool. And they're always covered with, like, really interesting stuff. So
the tails are always gnarly. The ribs are always gnarly. Any of the spines, any of the toes, you usually find weird little growths. We've got a great little pinky digit in collection, sadly, that's just all bulked up. And so it should be this long, slender thing, and it's just like black.
Yeah, I wonder if it has something to do with the fact that they have to, like, grow up so fast, right? Because I mean, that would definitely make sense. But yeah, they're super cool.
That was one of our previous talks, was how fast a little baby Parasaurolophus was growing, and it's fast. I would not want to go through those growth pains. No, thank
Well,
you.
yeah, I mean, if you don't want to be a snack, those are your options. So
Yeah. All right, so one of our questions that came up, an interesting one. So with ceratopsians, you get a ton of different shield types and horn types and nasal bosses and all kinds of fun stuff. And we have a question on, would Tatankaceratops be a triceratops with a deformity or unnatural bone growth?
oh my God, this is where I'm going to start showing that I actually don't know my dinosaurs very well. So a good way to find out if it's a deformity or a normal bone growth is like, how many of that specimen do you have? Is it well known to be that shape? Also, if you're talking about deformity from like from birth, then a scan or a sectioning won't help you because it's going to look like normal bone all the way through. But if it's like a deformity from an injury, then you could scan it and you can always find out that way. But I'm not sure, to be honest, because I don't know that specific animal. Sorry.
Yeah, I'm not sure how many individuals of that animal have been found, so I don't know the answer on that one either.
I think someone just said, I think there's just the one skeleton. So I don't know. Good question. I wouldn't bet my taxonomy on like a pathology or a deformity, I gotta say, because I mean, they're not that common, and if there's enough other characteristics for that animal, then I think that would be a valid taxon in that way, but I don't know.
All right, did skin grow over the bumps on the bones so that you could see a bump through the skin?
Ooh, good question. I think that depends on what bump and lump you're talking about. So for example, sometimes what we're seeing is like a small bump on like a bone, for example. But then actually there's a lot of swelling and there's a lot of like muscular destruction or whatever on the outside. And the effect on the bone was very minimal, but the effect on the soft tissue was a lot. And in that case, the outside could look really, really deformed, especially when it comes to like trauma, or if it was like a bite wound, for example, then a lot of soft tissue disruption would have happened. And in that case, you would probably see a lot of it on the outside. If you're talking about something like cancer or like that turtle cancer I was describing, when I looked up like instances of human osteosarcomas, the bump was actually very, very not noticeable. Like unless it was a really, really large osteosarcoma on like a joint or something, they're almost not noticeable. And a lot of cancers don't actually cause a lot of swelling. And so then in that case, you're not going to see it externally at all. So it just depends on how much of the soft tissue is disturbed and how close to the surface the bony bump is.
How many bones do humans have?
I think it depends on what age they are. So I think humans start out with like over 200 and something bones, and then you lose a bunch of them. And by lose, I mean like they fuse together and they count as one bone rather than a bunch. I remember I had a talk once and I said, oh yeah, humans lose a bunch of bones as they grow up, and people freaked out. They're like, where did my bones go? I'm like, they didn't go anywhere. They just fused into one thing, I promise. Like you don't just lose bones. But they didn't like me for that. So now I make the
point. Jeff, that's a carpentry thing.
Oh no.
Yeah, I think we start off with like close to 270 or something when we're born, and that's because a lot of people forget like on any of the long bones, so not just your arms but your fingers are also considered long bones. You divide up that bone into the middle portion, which is the shaft, and the ends, which is where most of your growth is taking place. So that's 3 in each of your long bones.
Yeah.
And then they fuse.
Yeah, no, it's crazy. And then and the skull just looks like a bunch of little parts. It looks almost like a fish skull at that point. You're like, oh god. But yeah, but then eventually they all start to fuse up. So it just depends on how old the person you're asking is then.
What are the limitations of CT scanning a fossil compared to cutting a fossil to view it in cross-section?
Ooh, good question. The biggest limitation would be how big your fossil is. I mean, it's a limitation on both sides. So if you have a really big dinosaur or something and it has a growth on it, your resolution when it comes to CT scanning will usually go down the bigger the scan is or the bigger the animal is, or sometimes you just won't find a CT that's going to fit your animal, so that's kind of problematic. But in general, almost always, if you're just looking for the best image, histology is always going to be better, because sometimes you can even see color differences that don't always relate to density differences in histology. So they can show you like where a certain amount of woven bone stops and a certain amount of dense bone starts. And even though technically they should be different densities, because they are, woven bone is much looser and dense bone is, well, dense. But because of fossilization, sometimes, you know, you'll have a lot of dense mineral that actually fills the woven bone, and then they become almost equally transparent, and that's not very helpful. But in thin section, you can actually see that in a color difference. So if you're looking for that level of detail of like, what are the cells doing? What are the surfaces doing? Are the two tissues really going to attach to one another that way? Then you really want histology. If you're just looking for tissue patterns like, is woven bone present? Is dense bone present? Is there erosion or is there no erosion? Then CT is usually okay if you can get your fossil to fit. I work on usually smaller stuff, so I'm okay, but good luck.
So if you're curious, for those in our audience, if you're curious again about the woven bone or that crystalline structure in there, go check out the histology video, because there are some close-up images showing woven bone as well as crystalline structure within those cells or within marrow cavities. But it's a completely different color than any of your surrounding bone. And you can see that in the histo slides. So go check out that histology video we had already done.
Woo, histology.
Do you know whether dinosaurs had epidemics according to the bones? So like with your modern creatures, like your Tasmanian devils, they're renowned for having diseases running rampant.
That's a really good question. I don't think we have that kind of resolution yet. I know that not a dinosaur, but a lot of like rhino fossils have arthritis, but that's just because rhinos can live with arthritis. There's a lot of that kind of stuff, but I just don't think we have the kind of resolution, because for you to recognize an epidemic, you would have to have a really good number and a big population, right? So let's imagine that all the Tasmanian devils right now fossilized, all of them. You would have to have a bunch of them to actually realize, oh, that's not just the one disease. Oh, this is a systematic problem. You would have to have a bunch of them from the same time period that haven't been moved around by a bunch of rock and earth, that haven't been spread all over the world, and that are semi-complete so you can actually diagnose it. So I don't think we know of any epidemics that way, unfortunately.
Do you ever see any bacterial or viral disease-related bone damage in fossils, similar to finding syphilis damage on human skeletons, or diet deficiencies like dinosaur rickets?
Good question. So for the viruses and bacteria, yes. So the vertebrae I showed earlier, the one with the weird metabolic disease, the metabolic disease is actually very similar to a human metabolic disease called Paget's disease. And even this disease is quite mysterious in humans, but one of the main things that people think it's caused by is a previous viral infection. So something happens in a human's life, they get this viral infection, it basically messes with the way their cells communicate, even after they recover, and then later in life they develop this disease. So when we published this, we called it a Paget's-like disease, because we can't really tell, you know, if it's the same virus, excuse me, that caused it, or anything like that, because, you know, viruses mutate so fast that it's very unlikely that you would have the same virus from the Permian that infected this reptile that would also infect humans. But it is very possible that it's from a virus. Also, we get all kinds of bone infections. So there's all kinds of diagnosed osteomyelitis, which just literally means like bone inflammation and bone infection, and that can be from all kinds of bacteria. So any bite that's been infected, any injury that's been infected, that is evidence of bacteria or virus or whatever, most likely bacteria, but that was there at that time. So we know that there is those kinds of infections. What was the other part? What was the other part of the question?
Dietary deficiencies.
Oh, yeah. So dietary deficiencies are interesting because So in humans, for example, when we are growing and for some reason, whether we get really sick or there is some sort of dietary deficiency, the bones grow abnormally. And in x-rays, you can actually see it. There's like extra lines in your growth plates, which show that you were either starved or you were really sick as a child. We don't really know what that would look like in a lot of these animals. And also, I think because social animals like us humans, we help each other out in such a way that like we make sure even the sick survive. I'm not sure if that would actually show up in other not so social animals, because usually if you're dietary deficient, it's either it's a really bad season and everybody dies, or you're sick or something in your body is not absorbing something properly and then you die. So we just don't see those kind of things as much in wildlife, in like modern wildlife, and I don't know of any dietary deficient stuff that we've seen in the fossil record, but I could be wrong. I think there might be something with eggshells, actually. I think there might be some evidence of temporal stress in eggshells. So because eggshells can actually grow in the body for a while, the parent or the mother would like be undergoing stress and not dying, but it would show in the layers of the eggshell. I can come back to that, but I think there might be something in eggshells.
What is your favorite fossil pathology?
My favorite fossil pathology is always something that's going to confuse me. And right now, I'm pandering a little bit, but it's the jaws of Tyrannosaurus, so like T-Rex and whatever. They have these holes in their jaws, and the holes don't line up to anything. And all the older individuals keep having more holes. And one of the theories is that it's this protozoan that is in water and it affects birds. Birds also get these holes in their jaws. But I'm really not sure because it doesn't look exactly similar, blah, blah, but that's one of my favorites because it's on such an iconic animal that you can just point to and be like, hey, you see those holes? That's a pathology, even though it doesn't look like an angry, weird bone malformation. I think that's always really fun. I think there's another one, let me think. Broken ribs are always good because they're really easy to point to, and then you can just poke someone in the ribs and be like, see, like that's what's happening up there. So that's always good. I do that in my museum all the time. So I think those are my favorite.
We do the same at our museum too. We have a big Highgate Mastodon that's on display, and you look at it from one side, it's this big, impressive animal, and you're like, come here to the other side, and you go around the animal, and you're like, see that? And they look up, what's different? It's like, look at these ribs, and there's 3 ribs in a row, and they're broken, and they've just started to grow the callus, and then it stopped. So you can see the callus, you can see the break. It's really cool.
Oh, that's awesome. That is awesome. Yeah, I mean, mastodons they always like just get a bunch of pathologies. And in general, like mammals are really fun to find pathologies on because it must be their physiology, the fact that they're just like warm blooded, but they take pathologies to an extreme, especially ungulates. Like I go through the modern mammal collection at my museum, is like one of the best in the world. And I'm just always looking at the weird teeth that they have because they're always chewing like all kinds of grit, breaking teeth, but they don't care. They keep eating and they get like horrible, horrible teeth pathologies. But they're fascinating.
You can't escape this one. What's your favorite dinosaur?
I think I've been, what did I say last time? I should be consistent. I think I said Camarasaurus because they're really cute. And I've only fallen in love this past summer, so you can't get mad at me. But yeah, they're just so like bulldogishly cute. And I don't understand their like proportions. So I saw the cast of the baby and I was like, what? Like it's just big body, small head, and it was just hilarious. So I think they're really cute. That's what I'm going to go for.
Well, it's like when I started paleo, we had very limited choices for what dinosaurs were out there. And so it's like, oh, I like Triceratops, and that was my number one. And then it's like, had you asked me now, like if we had all the choices now, it's like, oh man, hard to say. Like instead of a Triceratops, maybe I would have said like Regaliceratops or something. I don't know. Yeah, your favorites can change. It's okay. No judgment zone.
I appreciate that. Yeah, there's so many options now. And like, kids know exactly like the newest ones, or they know all the newest like Chinese dinosaurs. So when I do like outreach at my museum, they're like, "What's your favorite dinosaur?" And I'm like, I don't know. And they tell me like all these new Chinese. Oh, this one came out last year. So, I mean, I'm always humbled. So I'm open to change.
We have to keep up on all the kid literature and like all the dinosaur shows, Dinosaur Dan, Dinosaur Dana, the Dinosaur Train, all of that, because we will get questions on like Jurassic Fight Club was a big thing for a while. And so I had to keep up to date on all of the episodes because we would have tours and kids come in. Who would win in a fight? Velociraptor or Utah Raptor or whatever. You have to stay up to date. Otherwise they will call you on it.
I'm realizing that. I thought I could be like the too cool for school, like paleontologist. Oh, I was saying, no, it doesn't work that way. I'm realizing.
This was one that we're probably not going to be able to answer well, just because of how the disease works. Did dinosaurs have rabies?
Ooh, good question. So rabies is a viral disease, but it's really mostly affects the brain and like the sheath on like the myelin sheath, basically. I don't know how to say that in a simpler way, but basically your neurons are covered by a special kind of fat, and it affects that and almost leaves no like marks on the bone. So any disease that doesn't leave any mark on the bone or doesn't affect bone growth, we're never going to find like evidence for it, unfortunately. Yeah, so no. Although a rabid dinosaur is terrifying.
No, thank you. You need foam. Where's your shaving cream, Jeff? Let's see. We have our dinosaur bones hollow.
Ooh, yeah, yes, they are. Actually, most bones are hollow to an extent, unless you're like a deep diving animal or like a whale or a seal or anything like that. Most bones are actually hollow to an extent. So they have a marrow cavity. Yours are too. That's where we keep our marrow. That's where we make white blood cells. That's where I think red blood cells start getting made. So like a lot of things happen in your bones. Whether you mean like super hollow, like birds, then yes, absolutely. I mean, modern birds have extremely air-filled bones. They have like, basically think about it as if you want bone, but you also like want to be the lightest thing possible. So you just pump your skeleton full of air, and that's what it looks like. So yeah, they're super hollow. Yeah, most modern dinosaurs, aka birds, have really hollow bones, except again, things that want to dive deep in water. So penguins have quite dense bones. As for extinct dinosaurs, again, it depends on like how much weight bearing there is. So like sauropods don't have extremely hollow bones when it comes to their like some of their limb elements, but in their vertebrae, they're made of air, basically. I mean, it's like 90% air and then throw some bone in there. So it just depends on what animal and what region of the skeleton. It can actually differ quite a bit. So short answer, yes.
I saw you had a photo in there early with Matt Wheedle. How do you know Matt?
Oh, how did I unfortunately get to know Matt? I know him through Brian Eng, and when we got to go to Utah, and basically I asked him a bunch of vertebrae questions, and we kind of like mash up around the vertebrae stuff, and he always asked me, he's like, can you tell this part from histology, and can you tell this from histology? And that's kind of how we know each other. But yeah, he dragged me out to Utah in October last year, and we got to pull out that brachiosaur humerus, which was awesome. So yeah, that's how I know Matt.
Okay.
You'll be excited to see how your puppet comes along with Brian.
Yeah, but I think it's gonna have to end up probably being pushed to mostly next year. But I'm really quite excited. We'll keep the updates on social media. So
it'll be worth it.
Yeah, I'm really happy that it got funded. So that was by the Paleontological Association. So it was awesome.
I'm having fun watching his life-size Dilophosaurus reconstruction currently.
Yeah.
Yep. All right. Well, thank you so much for joining us. And yeah, have a wonderful time overseas. It's a little bit nicer weather than we're having today, hopefully. Although it's not too bad here.
All right. Thank you so much. You guys have a good day.
You too. Thank you very much.
Take care.